So with the tutor, I felt more comfortable talking straight because, you know, they're not thinking the test. So I was like, all right, cool, but let me talk to you guys. Am I sharing? That's fine. I think we're recording. All right. All right, let me come back to this one. And let's start with the second part. That's what we're talking about right now, and then we'll we'll go back to the stuff. Okay, so for the DNA and the amino acids, it's kind of the same thing. How much I expect you guys to kind of know. So you don't have to draw it. It's probably going to be a little bit more like the homework for that week. Right? So I think if you guys go on the Google Drive where you guys downloaded the assignment when we were doing it in class for week 2. You guys remember this? So I tried putting the keys up. I know this one's live. I'm not sure if the one for the first in class is live, but if you guys take a look at this, this is kind of like how much I expect you to know the DNA themselves, right? So remember the definitions of like, hey, what's a nucleotide, which is all 3 of the fragments? Oh, this one that's better. Like this guy, right? So nucleotide, we have the phosphate, the sugar, and the main carbon base, right? For the nucleoside, it excludes the phosphate base, right? So you draw these two. You know, we're talking about the sugar phosphate backbone, then it's just these two guys. That makes sense? Yeah. So can you like, so I will ask, I could ask questions about, you know, this kind of stuff, about structure. You want to draw them, it's just, can you identify them, right? And then the bases themselves, this is kind of at the level that I really want you guys to know. You don't have to know like which one exactly, like where exactly does your nitrogen land, or where's your carboxyl group, or carbonyl, sorry. What I will ask you about is more like this. Like you guys can see that the pyrimidine, right, it has just a single 6 member ring. And for that, the ones you're going to be using is cytosine, uracil, and thymine. Right, so that's where I do my thing that helps me remember. It's you build a pyramid, you cut one stone at a time. So then pyrimidine, CUT, and one circle, like that, one stone. Then the purines is the other way, which is now we're gonna have 2 rings, 1 5 and 6. In my head, as long as you remember you got 2 rings, and then add it in 1. Does that make sense? So you don't have to be able to draw them by memory, just recognize them when you see them at this level. The DNA itself, again, same thing. I'm not gonna make you draw it. I just need you to know what they're labeled. And the other one for that one is you should know the important connections, right? And for the DNA, we normally study it off the sugar. So when you're looking at that sugar, you gotta be thinking about this. There's an oxygen here. Then you have your first carbon, your second carbon, third carbon, fourth. This connects this way. You have the 5th carbon hanging out here, right? When we talk about the DNA, the 3 that matter is going to be number 2, right? What does this one determine?
DNA or RNA?
Say
RNA, DNA.
RNA and DNA, yeah. If you have an H, you know what are we talking about?
DNA.
There you go. Now?
RNA.
So what if the second carbon gives you this? What is the third carbon? Oh, so the third carbon, what does this one work with?
It attaches to the oxygen on the phosphate backbone.
Yes, sir, exactly. This would be the phosphate group of the next molecule. This is where you basically make a linkage spot to make the chain keep growing, right? And then the other important one is number 5. And this is where you have, what group do you have there? Z? Wait, where's Z? What group is that?
The phosphate group.
Phosphate group, yep. PEL. So in this one, I will pick on you because of this. Just knowing those 3 spots, right? So you gotta know number 3, DNA RNA. Sorry, number 2, DNA RNA. Number 3 is the linkage to the phosphate group. Number 5, both the phosphate group. That makes sense. Your order goes out. Does that kind of make sense? Yeah. So they'll go into transfers themselves. Same thing like that. We go, who started studying their amino acids? Everybody? Yeah? You wanna draw one? Yeah, you got this, come on, let's go. All right, so, I want you to pick people on the right-hand side of the class, over there, you can pick whatever you need. So, for example, to draw an amino acid, what are the 5 parts that an amino acid has? R group. You got one, R group,
Amino group.
yes. Carboxyl, all right, let's go down and see, we got it, what's your amino group, remember those? You don't have to draw this for the test, I'm just make sure you get that.
I know the name,
Your amino-
but there's a one with an H.
Nitrogen,
There we go.
and then 2 hydrogens connect to the nitrogen.
All right, and then, what else you have? You see your carboxyl? Yeah. I won't make you do this on the test, but it's good that we know. All right, carboxyl, and then what else do we have?
R group.
And then that final thing?
Hydrogen.
Hydrogen, yeah. All right, so that's your base for an amino acid. You won't have to draw them in the test, but you would have to recognize them, right? So, let me see. It's all right, go put the labels on these guys.
Do we need to know about all the, like, different types of bonds, like, between the different macromolecules, like peptide bonds?
Wait, let me get to that one, let me finish this real quick and then we'll jump over. All right, so let's say I told you to circle the amino group. This is your amino group. So can somebody open up the slides to the functional group? Let me see if I got them. So this guy's right. So we're talking about the amino group. There, we're gonna be looking at what kind of jumps off from that first carbon we really care about, like your alpha carbon. Whatever's jumping out for that would be, in this case, the nitrogen and the 2 hydrogens, right? So if I say circle the amino group. Yeah, circle now, yeah. All right, so that's what you're aiming for. Does that make sense? If I say, give me a square on the carboxyl group.
Is it both of those or just
So for the carboxyl group, you're looking at the carbon and all the oxygen groups with it. So, right, so that'd be your carboxyl group. Is that making sense? I believe in that PowerPoint for that, I think it's the next one after this one, but you can kind of see they'll do this kind of coloring. You can see your amino group is your NHs, your carboxyl group is your C with your O's, and then your other group is going to be your R group, which would be just the top one, right? So for that one, there are 20 amino acids, but I'll show you how I kind of want you to start hunting them down to figure out the 4 different types. You don't have to know all the structures, just know how to identify the types. But yeah, there was 2 questions. What was yours?
Is there a difference between on the carboxyl group, the single bonded oxygen? Is there a difference between writing the O negative and the hydroxyl group? Or is it the same thing?
It's the water's pH will change whether you have a hydrogen or not, right? Yeah. As long as you draw C double bond O with a negative charge, because you're missing that positive charge on the hydrogen, that drawing is correct.
Okay.
If you draw C double bond O and an H with no charge, right, because it cancels out, then you're correct.
Okay.
Yeah, so as long as you do one or the other, you're okay. Does that make sense to everybody? All right, cool. Boom. 1, 2, 3, 4. So in your notes, you guys, amino acids, five groups, right? And I'm bringing this up because the important ones are basically these 2, right? Because remember, if I told her, hey, draw the next amino acid here, or if you remove the next reaction, it would show up here, and then the nitrogen, the amino group, is the one that links up to this oxygen, with that H, and give it a water, then you up, and you have your next amino show up. Yeah, so that's kind of like for DNA, that sugar phosphate backbone. like this, where you have that linkage from 5 to 3. In amino acids, the connection is you start at N, it goes to the carbon, right, your carboxyl group, and here you show up the next amino acid like this. And carbon, carbon, like that, right? And here would be your next R group.
It only has one H off of it, right? Or would it have two?
You're on one. So nitrogen, the only thing I remember from the whole chemistry part is nitrogen needs 3 partners, 1, 2, and that's your third one. So you're correct. You're not gonna have to draw that either, but I just want you to remember, like, that's the connection on how you're making the chain. And then we have to treat the R groups, I'm gonna treat them a little bit differently. Yes.
Can you explain what the R group is again?
Okay. Let's see. Before I just keep going, DNA wise, are you guys comfortable with what you have to learn for the DNA? The chain, and then the whole pyrimidines, the bases, and the number of rings? Yeah? All right. Let's go to the polymers. I think the question was to go over how we make the reaction. So I'm gonna go back and draw a little bit more before we go to the R groups, yeah? So one more second. So in this case, I have the slides on my drawing stuff. All right, that's what we have on the board right now. So here we go. All right, so for this one, there's what Cody, no. Yes, Cody drew it, right? You have a first amino acid, it starts from the end. There's his alpha carbon, there's the carboxyl group, right? The 2 oxygens. There you can see that this oxygen is gonna steal the hydrogen from this amino group, right? So it's gonna take 2 hydrogens with it. If one of the hydrogens left behind, like, excuse me, what was your name again?
Morgan.
Morgan said, right, there's an H that stays behind because there's 3, 2 left. 1 stays with that nitrogen, and then this nitrogen links up with that carbon. And that's what you're seeing there. Yeah. And for this one, the main thing to kind of remember here is the orientation is specific, right? We start at the N terminus, and then they call this the C terminus. So we have N, this is your first amino acid, C, N, your second amino acid, C.
So is that the glyco something linkage? Is that what reaction's happening, glycosidic?
So the glycosidic linkage,
Or is that only between polymers and monomers?
so glycosidic, that is when you're talking about sugars.
So this is a dehydration.
They're both called dehydration because you're both, in both cases, you're taking out
Yeah.
water, right? So that's the process of taking out water is dehydration.
Yeah.
Because you're gluing proteins here, it's called a peptide bond.
Oh.
In the other case, because you're gluing sugar, then it's the glycosidic linkage. Yeah. Everyone comfortable with this? Yeah? All right, cool. So for this one, this, let's see, make sure. So when we're studying these, to me, both of these go together. Or this is where you kind of, they come back up, right? So for the first one, let's start it backwards because they're the easiest one to remember, right? For the proteins, the R groups, I just want you to know that there's 4 different types, yeah? And what I kind of, the way I remember them is that I look for specific functional groups, and those will be my clues to what type they belong to. Yeah. So if we start, you guys started making your rules for this, guys. The first one we're looking at, let's look at this, right? Or my favorite one is the acidic. If you have something that's acidic, and what's something that you guys see in the R side of the group, right? You see C, double bonded to O, and then single bond to O, that carboxylic acid, right? So if you see C, O, this is negative charge, yeah. If you see this, you know you're working with an acid. So whether it has one carbon in the middle or two carbons in the middle, as long as you see this show up, you should, in your head, you should be thinking that amino acid is an acidic, has an acidic, carbon, right? Let's look at the next one. We're looking at basic, right? What are we looking at? What are we seeing, right?
Nitrogens.
That's right. So you can ignore the carbons. What are we seeing? NH3. Plus, you're seeing, here in this case, what I look at is we see that we have two nitrogens stuck to a carbon, right? So you see, there's a carbon there, NH2, and NH2 plus, right? And here I see that NH plus sticking around, right? So we have NH plus, carbon, and carbon. But what you're looking for is this NH plus, NH plus, NH plus, NH plus. That's my basic stuff. Then we have, the next one is gonna be our polar group. Right, what are we looking at? We're kind of, hunting for the polar group.
Oxygens.
That's right. What's the difference between this polar group and the acidic?
So the acidic has a carboxyl acid.
Yes,
and
sir.
the polar is not.
What's OH called?
Hydroxyl.
There you go. So hydroxyl is one of the ones you're gonna see a lot, right? 1, 2, 3. Even though it's after a ring of carbon, doesn't matter. As long as this is at the edge, you know what we have, right? 1, 2, 3. And so OH is one of the patterns. If you see a carbon going to a loose SH is another pattern, right? AKA my favorite amino acid. I don't know who asked this, but you know, this is the one that helps fold up your protein, so it's super important. SH, and then the other one is this double bond O, or the NH2. Right? If you see any of these three markers, you know we're working on the polar side chains. Yeah. And then once I do that, sorry, polar hydrophilic, HP, right? Then we get to these. So these are, remember, they're the boring ones, or stuff is hidden, right? Hydrogen, boring. Methyl group, right? That's the only one that's non-polar out of all these. It's the methyl group. If you see carbon with hydrogens, it's gonna be on the non-polar, and it's gonna be on the hydrophobic side, right? Carbon, 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 carbon. Only one nitrogen, so at that point, if it doesn't, like you see here, we're looking for like those NH3 pluses or the charged ones. If it's not charged, it's gonna be non-polar. Non-polar, same thing. We got carbon. Here, there's sulfur, but it's hidden inside, so it's still gonna be acting like a non-polar. A chain, a L shape, like a methyl group on the side of the chain, non-polar, methyl group at the end, non-polar, methyl group, the short version, non-polar, right? So if you just see carbons or just sticks drawings, in your head, you should be like, this is boring. It should be non-polar and hydrophobic. Yeah? So you won't have to draw them, but I do want you to recognize them. So this will be in my head, this is like my cheat sheet that I'm thinking about when I'm seeing this. Yeah. So do you guys feel comfortable picking these out, identifying them? All right. So here they're color coded, but imagine I took everything away. Right, the purple's gone, the yellow's gone, and I give you this amino acid. Right? If I tell you, hey, tell me what it falls into, that's why it's really important for you guys to know what the back chain is made out of, right? Because if you look at any of them, what are the two things that we're gonna find? We're gonna find an amine group in the front, which is gonna be an H3N plus, which makes it look something like this, right? But since it's not the side chain, it's not gonna matter. The other part is on the right side, you have the carboxyl group, C double bond O with O, which would make it look like it's an acidic part, right? That's where your amino acid comes from, the name itself. Don't get fooled by these parts. You're like, hey, once you find an amino group, a carboxyl group, then you're wondering from this central alpha carbon, what's going on above? And you use this table to kind of get you to pick what group it falls into. Yeah? All right. That answers most of the question on this side, but I believe Morgan's question was still left behind.
You answered it with the drawing,
I did.
with the peptide bonds.
Okay.
You're good.
I feel like you said polypeptide. Maybe I was wrong.
No, I was just wondering if we like needed to know the different types of like linkages between the macromolecules. And like the glycosidic linkages, but you answered that.
Okay,
So
yeah,
you're good.
okay, perfect. So there it's getting rid of water, it's called dehydration, but then based on what you're gluing, you give it the name of what you have, right? It's either proteins for peptide bonds or the sugars, yeah. All right. Cool. All right, so for this part up here, I think I answered it pretty well, right? I feel like you should be comfortable with the nucleotides, with the amino acids. You don't have to memorize the bases themselves, like each position of the chemical, but how they're arranged. And for the amino acids, it's more about, can you pick out the functional groups to figure out what that amino acid is? Then we have know about pyrimidines and purines. Yeah, you should know your pyrimidines and purines. Tell them apart just by the ring number and which bases fall under it. Right, so that's where I do my pyramid thing. So pyramid, cytosine, uracil, thymine, okay. And what do we know about peptide bonds? I think I answered that pretty well. And polypeptide formation. So I think, yeah, we covered that, we should be fine. So now let's go to question number 1. Right, so this one was part of the essay that was due today. Since I forgot to remind you guys, we're gonna move the deadline to Friday. It's a quick 1-page, just bringing up, I think it was emergent property. So an emergent property, like who can define emergent property? From my right side back there. Yes, emergent property. Yeah.
Me?
Yes.
You whip out my notes.
Do it.
Emergent properties, okay, approaching the whole to understand the parts, like thinking what's a cake made of, and then you go through the specific ingredients.
Exactly, right? So it's like, out of the wait, sorry, say it again. The whole,
Like approaching the whole to understand the parts, so like approaching a cell to understand like the function of each organelle.
That's backwards, but this is, yeah, so it's like, how does understanding the components that make up the cell end up coming together to give you that extra property on top?
I thought that was
It works.
reductionism.
Hold on, let me see. Yeah, so reductionist approach is kind of how you said it. You grab, like if you want to study a cake, you start breaking down what each one can do, right? For the emergent properties, like kind of what things comes together as you put it together. So when you bring in the flour, temperature, the sugar, the yeast, all those come together, they give you that final thing, which is going to be the cake.
Yeah.
Yeah. All right. So now that analogy, I need you to explain to something outside of this class, not a cake.
Not a
But
cake.
not a cake, something else. It could be anything. So that's gonna be your guys' essay. So have any ideas? It could be a machine, it could be people, relationship, anything you can think about of getting the idea together of how do small things come together that because of the way they function or interact, they end up building up the bigger thing. Yeah. It's informal. I don't need resources. You could just, throw in ideas and then we should be good to go. But bring up that idea that when you bring up understanding the parts and as they come together, they give you something extra or something that the parts themselves couldn't do. Yeah, yes.
So basically the difference between reductionist and emergent is that the reductionist focuses, like it says, on the isolated components or the specific parts, whereas emergent focuses on the broader properties that come out of the parts.
Exactly. So normally when I have this table, I would, like in my notes, I literally have arrows, right? So it's like we're going from really small things like atoms and molecules. And what we've been doing right now is just basically keep going up, right? So we understand how small things like I've been going through organelle after organelle after organelle with the whole thing of like, once you understand how each part of the organelle comes together, we can build up a cell that has a whole big function, right? So that's basically you're going kind of up in sizes. That's the way I see it, right? And then, like you said, the other case would be we would go down and it'd be like, we would have to then break up each single part where I'm like, hey, how does a microtubule work? And we talk about, hey, the microtubules, one end grows really fast, one grows really slow. And based on that, we can trap one of the ends or give like stabilize the other end to change its dynamics. Yeah. But you're right, it's the thing that we're focused on our study, right? Is it, if it's the small thing going upwards, or if it's the big thing and we're going down in size. Does that make sense? No? I see lost eyes. Okay, I see a few head nods. I'm gonna take it as a yes. So give me that paper. I think in here I did give emergent and reductionist approach. Let me see, emergent. Yeah, so emergent, I did talk about the microtubules. That's the thing we're talking about right now. So when we combine three things, the microtubules, remember those are those cytoskeleton elements that build up to build the straw. The other thing that we have is going to be motor proteins that can move along that straw. And then we have the vesicles, the fat bubbles, that basically they can encapsulate things. Once you put those three things together, this is where you get the thing about how you can actually traffic proteins, right? You have the microtubule, which is going to guide you where you're going to go. You trap your protein inside the fat bubble, and then your motor protein can literally walk that fat bubble over to where you need to go. Yeah. And then the reductionist approach is going backwards, which would be like if the thing that we're trying to study is the DNA, we then try to break it down into what the DNA is made of, right? And then that could be like, hey, what's it have? Sugar phosphate backbone. Then you have your different nitrogenous bases, and then we're like, oh, each base has a binding partner that helps us with replication, blah, 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 blah. Yeah? All right, so I need some of those coming in, yes.
Would the nitrogenous bases on like an RNA sugar have the same, like would it still be like guanine, thymine, all that?
There's one big difference. What's the big difference between RNA and DNA with the base?
RNA has uracil and it doesn't have thymine.
Correct. That's the only big difference. From the nitrogenous bases.
Yeah.
Right, yeah, from the nitrogenous bases. All right, and then how much do we need to know about isomers for the midterm? So the isomers, I didn't go that much into it. So for the isomers, it's kind of talking about when proteins are being, or when carbon chains are being made, they have, remember, 4 bonds they can have, right? And they normally set up in this like tetrahedral pattern, like there'll be 3 in like this going down, and your 4th one will be sticking up like that. So imagine this would be the central carbon. My fingers would be the 3 things it can bind to, and 1 points down. The thing about the chirality is because the molecules can't like move like this past each other, it has a certain like a certain positioning, like where if I move clockwise, you can see it with thumb, pointing finger, and middle finger, right? If and my arm would end up pointing that way. So this my left hand. If I have the same 3 atoms, right? My thumb, my pointing finger, my middle finger set up like this, I can't move my hand in any way that'll make all of them match up, right? Like if my arms are matching, my thumb is matching, and my middle finger is matching, you can see that technically my pointing finger is aiming in different directions, right? If I make my middle finger try to match, like I can't get them to set up in the same position, that's your chirality. Because like I said, the molecules can't shift through each other. I didn't go that much into it, but it is important. I'm not gonna bring it up in the, I'm not gonna bring it up in the test, but because it's a 3D shape and I didn't want you guys to get the 3D models, it's really hard to explain it. The only thing to know, just for you guys on stage, is at the atom level, based on how they shape themselves, it'll be messed up from that point and it builds up. You can see it at that point. You can also get it for proteins. They'll change their shape, and then because they kind of popped into a position, they can't switch into a different one, or they'll have like different arrangements. And some of them are super messed up, like they'll mess up our I think our grain is one of the things they can mess up. And really with motor proteins, but we're not gonna bring it up, so it's about 3D conformations, but that one's too you'll probably go into it later. especially for neuro people, you'll visit her a lot. All right, and the other one is, do we have to know all 7 of the main functional groups and in how much detail? Yes, and I love them, so yes, you do. So, kind of like I asked you guys in class like this, right? So what's this guy? This guy? Yep, and then you have the C double bond L, what's that one?
Carbon.
Yep. Yeah, so know all of them like that. You should be able to see that when they come off a carbon chain. Make sure you include the all the elements, right? Like, for example, here you have an amino acid. If I'm talking about the sulfhydryl, right, it's the S and the H. Here, when I'm talking about a phosphate group, it's the phosphate and the 4 oxygens. So you make sure you encapsulate the whole thing. And the other thing to note here is that only the methyl group is non-polar. Everything above that way is going to be polar, right? All right, any other big questions? Because I think that's all from the review session form.
Do we need to know, like the different types of proteins, like support proteins and like, there's like 12 of them.
What do you mean?
The,
Like transport proteins.
yeah, like stored proteins, defensive proteins, like receptor proteins, like they're,
Like the functions?
or are we gonna have to know like examples of them, I guess?
No, I'm not gonna go into that detail. It's good for you to know that they can do all those jobs, right? But to be honest, I think later on in the text is when you go heavily into, we start actually looking at those jobs when they're doing it. So right now, they just gave you an intro of like proteins can physically move things, cut things, or they can bind to specific partners. Like they're super shaped and charged to make perfect binding partners, that's it.
Question about the exam, is there any multiple choice or is it all short answer?
So far, I got like 20 questions and there's a little bit of everything.
Okay, gotcha.
So it's gonna be like, sometimes like definition, where it'll be like matching answers. Then I have a section where it's just like, or part B, I call this short response. So it'll be like, I'm giving you like less than a quarter of the page. And it'll be, I don't want you to write a whole essay, right? So it'll be like, hey, if I ask you a specific thing, give me the like your most condensed, precise definition of it. And then I give me a quick example of how it's connected. Right? So be quick on those. And then at the end, right now I'm still working on it for part C. It's gonna be one long question. So you guys might have a choice between 2 or 3, and those I'm expecting like maybe like half a page. And yeah, those will be the long ones. So one of the things I was saying is like when I was thinking about is when you go through the test, I want you to look at how many points a question's worth, right? And it's like I'm not gonna expect you to write an essay for 2 points, right? So normally if you see a longer question, spend more like six points is maybe worth 6 minutes of your time because you know the whole point. The whole test is 50 minutes, right? If it's only worth 2 points, it should only be worth 2 minutes. Like don't. But yeah, and yeah, so I have the spaces reserved. No one's hit me up to say they want a specific quiet space or like a specific pre or post time thing. If you do, please reach out to me so I can start putting them in like who's gonna take what room and everything. And then I'll have the test printed out. And then you guys should be pretty good to go. And I still haven't checked it to see how clear it is about the question types. I think some of you guys gave me comments on the homework, right? Where you guys were telling me like, I didn't give you guys hints of like, hey, it's multiple answer or it's multiple choice or it's true or false or how the matching works. So I'm gonna try to be better at that on the quiz itself. Because in one of them, I think it was like, I asked you guys about microscopes, right? And I was trying to get you guys to think like, hey, what technique can you use? And it was like, one of the things I was trying to be exaggerated, but like for the elephant leaf, it's like this big, like it's a huge leaf, right? There, if you guys remember the drawing where we're talking about the microscopes, it's like a microscope, because it zooms in so much, you can only capture a really small portion of the tissue and you can't see anything else. So if you're trying to take a picture of something that's 2 meters big, you know, your microscope, you'd be there for like a year just taking pictures over and over trying to get into the whole thing. So for that, you can only use the light microscope. Sorry, the light microscope, not the light, your eyes are a camera, right? That's that magnification from kilometers to centimeters. It's kind of like where our eyes, I mean, mine probably at, you know, I don't know if I'm making centimeters now, but you know what I mean? Like it's limited, right? Then you shift over to the next window, which is, the second part was for an example of if we were looking on the surface of the leaf itself and we were looking at cells that are like microns by microns. And there I wanted you, there I kind of messed up, or maybe my question was too clear, was what can I use to image that? And if you look at the range for a light microscope and the range for electron microscope, they both overlap. Like you can image microns with both of them. So the second answer was you had to put like for that second sample on both light and electron microscopes, it would work. So that's why I was a little bit off. And then one of the ones that I messed up was, I think most of you got it right, was for the question talking about the mitochondria and the Golgi, right? So what does the mitochondria do?
ATP production.
ATP, yes. And then the Golgi? Transport, yeah, but I spelled mitochondria with an extra R, so everyone got it wrong. So I need to fix that right now. Yeah, but I was tripping out because it kept saying other, and I was like, why does it say other? And everyone got it wrong, and I was like, doesn't make sense. But yeah, so that should be good. All right, so I'll try to make it clear about those. The matching one should be okay. And I haven't done any fill in the blanks yet, but I think those might be a little, oh yeah, I do have a fill in the blank question. And it's like, it asks, like it gives you a story of how something works. And then I give two different types of examples. Or like how it works in different organisms. So when I put one organism and the other organism, and I'm expecting you to answer it like that. Yeah. All right, any other big questions? Small questions?
So what percentage of the test on Wednesday will be like from chapter 1 and like chapter 2? Since that's like an introduction to like what we.
So let me think. So chapter 1 was the unifying,
Like organization.
yeah. I'm trying to divide it pretty evenly.
Okay.
Yeah, so I feel like the first one, because it's a way to think or like how we appreciate bio, right? It's like feel comfortable with the themes, right? You should feel comfortable with them. Like information, what was the other one?
Energy and matter.
Energy and matter. See, that one I went a lot less over than the other ones, right? So when you're studying, I normally look at my slides, and whichever slides has the most sections for that specific topic, is the one that I kind of went into greater detail with you guys. So you can use that to help narrow down your area of focus. Like this question shows, right? Like it's like, hey, I didn't go over isomers that much, so it's like, I'm probably not gonna ask about isomers, right? Like there's stuff like cytoskeleton, like which ones are my favorites?
Microtubules.
No, it's sir. And then what's the other one?
Microfilaments.
Yeah, it's the ones I hate.
Intermediate filaments.
Fake filaments, guys, don't trust me. So yeah, so use that to kind of help guide you what you study. The chemistry one, which is number 2, that one, to be honest, because we're studying bio, we're not really like how often we're gonna bring up valence electrons going forward is gonna be rare, right? Or like the protons and the neutrons, it's not useful in kind of the end. But what are the stuff that from that slides that are super important that I'm always talking about?
Types of bonds.
Charges, right? The types of bonds. I'm like, it's a hydrophobic, it's a polar, it's a non-polar, it's a hydrophilic. Ionic is gonna come out later, but it's one of the ones where they are useful. So those are the things where I'm like, I am gonna squeeze that out of those PowerPoints. Knowing the mass of an electron, the magnification we work at, we kind of don't care. You know what I mean? Like it's like we don't care. Yes.
What else do we need to know about like monomers, polymers, polysaccharides?
Polysaccharides. All right, define all 3 to me.
A monomer is used to bond polymers together.
Not
It doesn't bond
It
them
makes
together.
like the monomers make up
Building
polymers.
blocks.
It's small subunits of.
So in relative terms here, Each one of you in my class is a monomer, right? If I put two of you together in a group, it's a dimer, right? And then if I link up the whole class, then that's considered a polymer. Yeah.
Yep.
That's pretty much what you need to know. Cause that's an idea of how things link up, but in reality, you know where I'm gonna push you guys, right? I'm gonna be like, hey, amino acids, monomers, this is a monomer, here we have a dimer showing up, but give another one, now we have a polypeptide or polymer. Same thing with DNA, right? All right, let me see what else. Oh yeah, I was gonna show a slide. Somebody got me, I was like, I love this picture, it's super awesome. And then I was like, the one on the slides, I said I hated it. And then you guys put it up here and I was like, you guys are bullies. That picture up there, right? At first I was excited, I was like, this picture looks dope, I love it, because how I explain oxygen and hydrogen, right? But in the class, I said I did, and I was like, they got it wrong. And then when I saw it, I was like, fuck, it's wrong here too, right? And I'll look this close to it, I'll throw it in the test, but I don't know. So what's wrong with this picture?
Oxygen's bonded with oxygen.
No.
Yeah, that's what got me, but everything else I loved it, because I was like, this is what I'm trying to get across, right? That your oxygen and hydrogens, that's your polar covalent bond, right? That's like that part of like, it's one thing, relatively it's the stable thing. Then they have this second type of interaction, which is that cohesion, but if that like arm was coming from the hydrogen, you know, it would be dope. I might have to Photoshop it and then I'll make it look good, because I do like it. And then I also love this part, right? So this part, it's the same thing, where it's like, hey, you can stick to other stuff, but it'll do the same thing. They have to be polar for the interaction to happen. Yeah, so that was good. You guys got me, awesome. And let me think. So slide 3, no, lecture, what was lecture 3 about? Water? Yeah, you gotta know water, that one, everybody loves water. So water, with its hydrogens, electrons, I'm like I said, I'm not gonna go heavy into the electrons for water. What I do care about water is polarity and talking about hydrophobicity, right? Hydrophobic, hydrophilic, that type of interaction. But I've been pretty hard on you guys, like every homework, I've been bringing that up. And I'd be like, hey, keep telling me about it, keep telling me about it. So that's what I'm gonna push for you guys. Then, to be honest, it's like when you figure out all the stuff that it can do for us, like the important things, right? How it can bind to itself, how it can control temperature by taking up a bunch of energy and avoiding the temperature to rise, or it can freeze things, or when it freezes, it expands, so it floats. Those are like the big properties of what help us in life. So those are the things where I'm like, hey, make sure you feel comfortable with those. And the fact that all of them are coming from like this hydrogen bond, yeah. Lecture 5. All right, let's see this one. So for this one, again, it was a way to introduce you guys to the other ones. We've done carbohydrates very little, which are sugars. I've talked a little bit when it talks to the cell wall, or like you said, your example of monomer, or what's it called, monosaccharide, disaccharide, and the polysaccharides. You guys seem that have been a lot more heavy on the proteins and the nucleic acid. Yeah, so still go through it, feel comfortable with the other ones, know the definitions, but you know that the ones I'm gonna focus on a little bit more on is gonna be your proteins and your DNA, because those are the stuff we've covered so far really heavily. Yeah, I feel like the steroids, I like sprinkled it like it was salt on food, like it was just like steroids. They have functional groups, right? So go light on the steroids. The phospholipids, that one, what are the key things you have to learn about phospholipids?
Hydrophilic. Hydrophobic.
Exactly. 2 parts, know which one's the hydrophobic, which is the hydrophilic, and then know how it's super important to building shells around organisms, right? Around organisms and around, what are they called, organ systems or organelles, right? How to build shells around organelles, because they basically act like a sphere of a particle's, its charge is gonna affect, and its size will affect if it can make it through. And for the most part, it's gonna act like a shield. All right, number 6, the nucleic acids. Just know everything. I love nucleic acids, so I'm just kidding. I just know the stuff that I went over, right? Here is how the chain works, how to call it out, like how it's patterned, and then just know that there's 4 different types of bases. those bases, they give us some cool benefits, but it's like what we talked in the homework, right? It's information in the order they appear. Because of their hydrogen bonds, we can create copies of it. One of them is for replicating, the other copy is for your RNA to make your protein. So that ability to temporarily bond with hydrogen bonds is the whole reason why we're able to do so many functions at low energy costs, right? Because if we had to make and break covalent bonds to bind DNA, to make an RNA, it'd be super expensive for us. All right, DNA, RNA, phosphate bonds. I think I went over this one pretty well. Mixture rate for the proteins. Yeah, so this one, same thing. Know the 5 parts, the carbon and the 4 things that attach to it, and then be able to call out the groups. This, let me think, for this one, for the different structures, I feel like I went over it, but I don't know how in depth I went over it with you guys. You guys remember the primary, secondary, and tertiary stuff? Yeah? All right. So this one I think would be, I think definitions, I think we barely hit ordinary, or I would just do proteins working together. So normally it's, you kind of stick up here, like primary and secondary, which will be, primary is just the amino acids in a row from N to C. Secondary is if this backbone, right, interacts back with itself, it'll make coils, or it can make like it can make the, it's called data sheets. Yeah, data sheets off of this. That's pretty much as much as you need to know for those guys. All right, no notes on that one, huh? You guys are good? I think this is when we started talking about the organelles, right? So I started bringing up the different compartments there. Know the name of the organelles and know what they do, right? So I remember I was telling you guys, figure out your keywords for each organelle or that key function. So then feel comfortable with those. The other one is cells 2. What was this one? The cytoskeleton. No, this is the last one. I think the first thing was the membranes, then it jumped into like the organelles and how they each bring different things. You know, there I'm gonna love to put some compare and contrast stuff, right? Because there's where we brought up eukaryotes and prokaryotes, right? Eukaryotes, us, the fancy cells, right? We have all those different compartments, we're bigger, we hold our nucleus, our DNA in the nucleus, and then we have to work through these different membranes. Prokaryotes, they're lazy, they're just one single cell, they have everything in the mix. Makes it easier for them to just make our DNA right where the RNA is at. They have to make RNA right where the DNA is at. They can make their proteins all locally. It doesn't need to go through different membranes with different compartments. And this one, you guys already talked about it. You know my favorites on this side. Yeah, so these 2 bold, this is like lowercase bold. And then this is important because now we're getting into, this is everything inside of the cell. Or sorry, yeah, this is inside of the cell, and then here's the jump to outside the cell. So feel comfortable with these 2, right? Which is the difference between an animal and a plant. Remember, I love plants, but I think they went more into detail with this one. And these type of jugs are also important, right? They help communicate across cells. That's one thing. They also can function to help attach them to each other, which I think this is your desmosome. And then finally, your tight junction is, that's a function that they do together for the organism as a whole, right? That's the one where they kind of build like a wedge around themselves. They kind of act like a shield where no liquid will pass through any of the cell parts. And that's it. Start studying, go through the organelles. The big thing I want you guys to be thinking about is how are the stuff working together to build up the cell, right? Like individual. I'll see you guys here on Wednesday. I'm wearing my coat because it's battle time, you guys. It's gonna be serious. 55 minutes, and then you guys. I have office hours today after this for 2 hours, tomorrow morning from 9:30 to 11, Wednesday morning from 9:30 to 11. There you go.
What?